Hello, everyone, and welcome to today's AAPS webinar. Today's webinar is hosted by Somru Bioscience. You may submit your questions at any time by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. It is my pleasure to introduce today's moderator, Rafiq Islam, Chief Technology Officer for Somru Bioscience. Rafiq, the floor is yours.
Thank you, Stacy, and welcome everyone. Good morning and good afternoon and good evening to you, depending on whichever the part of the world you are joining from. We have an exciting topic for today's e-learning webinar. This is very close to my heart in terms of ultrasensitive bioanalysis. We also have two great speakers who will be presenting the case studies on utilizing ultrasensitive solution for HCP detection, biomarker analysis, and the biotherapeutic PK quantitation. Will, if you can move the slide for me. Just a little bit details on the webinar is we'll have three key portion. The first, we will talk about the platform, immuno-PCR, the workflow, and how it can combine the immunoassay specificity with the sensitivity of PCR. We will talk about how the platform is different from ELISA and ECL using multiple case studies. And then we'll close it up with how this platform can improve bioanalytical workflow to improve sensitivity and the compliance. Let's go to the next slide. So I have the distinct pleasure to introduce two of our speakers. The first, Waldemar Carvalho. He likes to go by Will. He is a bioanalytical principal investigator with Somru Bioscience. He has been in the industry now over 6 years. He has a PhD in chemistry from University of Alberta. And he is one of the key inventor of mPlato solution from early on. And Will, if you can go to the next slide. I would introduce both speakers now, and they will kind of tag team this whole presentation. And then obviously we'll be available toward the end to answer any questions you may have. So our second speaker is Jessie Ricks. She is the senior bioanalytical scientist and the principal investigator at Somru Bioscience. She leads our method development team for PK immunogenicity and biomarker. She also supports some of our analytical need in the area of biomanufacturing. With that, I will hand over to Will. Will, the floor is yours. Thank you.
Thank you so much, Rafiq, for setting up this stage. And thank you, everyone, for being here today to listen to this great topic. So here is today's agenda. Like Rafiq was saying, we're going to start with an overview of mPlato technology, then walk through real application examples, including PKA, host cell protein detection, HCP for short, and biomarker analysis, including TNF-alpha and L6. We will also touch on multiplexing and conclude with final key takeaways. So mPlato technology, it is a immuno-PCR technology based that was designed in a way that any traditional ELISA could be easily adapted to achieve high sensitivities. So I'm not going to go into much details on the technology itself. I have presented a couple of times here at the APS already. If you guys wanted to know a little bit more, you can contact me and I can share the link for some of those presentations. But basically in here, when we have a traditional ELISA that has a detection antibody that is bioethylenated. So in a normal ELISA, what is added is streptavidin HRP, so a conjugate that is used to generate the color. In here, in Amplato, we replace that enzyme conjugate with functionalized gold nanoparticles. So in sense, the immuno-PCR, the technology, it combines the flexibility of ELISA. So any target molecule you can use to detect if you have a pair of antibodies in there. And also the high sensitivity and exponential amplification power of PCR to enhance that signal. So in here, those functionalized nanoparticles, they act as a oligonucleotide carriers. So they have 100 copies of oligonucleotide on them that is later used to amplify and quantify by either qPCR or digital PCR. So in the normal, so this in here, that's the Amplato technology that represents the Amplato technology. So in the normal ELISA, like when you run in the assay, at the end, the DNA is released from the particles, and again, it is amplified and quantified by digital qPCR. So there are a few challenges, right? Historical challenges when using immuno-PCR. So the use of immuno-PCR has been limited by several practical and technical challenges. Like all the critical assay reagents, the nanoparticle quality and batch-to-batch reproducibility are essential to obtain reliable and reproducible results. And variability in the nanoparticle preparation can directly impact the assay performance. In addition, equipment compatibility can be a barrier as well. So immuno-PCR, you can perform it in normal ELISA strips, but also in a PCR plate. And many standard plate washers, they are not designed to wash PCR plates, which can make assay execution a little bit challenging. From a laboratory perspective, immuno-PCR workflows, they require physical separation between the immunoassay steps and the PCR analysis to minimize contamination risk. And in many labs, they are not originally designed with this segregation in mind. So we need some spaces that are dedicated for the PCR and the assay, and also some preparation for the reagents. Finally, also here, successful implementation depends on heavily on well-trained laboratory personnel. It is to minimize any contamination, source of contamination. So proper training and strict workflow control are critical to minimizing any contamination and ensuring consistency and high quality data. Oh, sorry. In order to overcome some of those challenges, we developed Amplato, right? So the particles, they undergo extensively quality control. This includes we looking at the oligo density on those particles. We try to keep it between 200 and 500 per particle. We also look at the streptavidin loading, the size dispersity, and the surface modification. So in here we have a graphic showing the UVV's extinction of the particles before and after functionalization. We also aside of characterizing those particles, we also perform some quality on the performance of the particle in the immuno-PCR itself, which include like batch-to-batch reproducibility using our standard immuno-PCR, and also we look at the stability of these particles using our standard immuno-PCR. So this slide here shows how the data looks like from the qPCR once the qPCR is performed following the immuno-PCR assay execution. So after running the samples in the instrument, we obtain amplification curves. And those curves are dose dependent, so they are concentration dependent, which reflects like the concentration of the analyte. So in this case in here, the amplification curves are shown as a function of human IgG concentration. So this is our standard immuno-PCR that we used to evaluate the batch-to-batch reproducibility of those particles. So as you can see in here, higher concentration of human IgG, we have earlier amplification, and lower concentration of the human IgG, we have a later amplification. So once the amplification data is obtained, then analysis is performed using a in-house tool. Importantly here is that the analysis approach is assay dependent, ensuring appropriate interpretation and quantification for each specific applications, because there are few ways that you can do the data analysis in here. So Amplato is being also adapted for use in both qPCR and digital PCR, which enables flexibility in how the technology is deployed. So this means that Amplato assays can be performed using either platform, depending on the analytical needs. And both technologies offer distinct advantages, which is listed here in this table. So for, I just listed 4 in here, in terms of sensitivity, the qPCR has a very high sensitivity, but it depends on the amplification efficiency. While the digital PCR is ultra high sensitivity and often superior for lower abundant protein due to the absolute counting. In terms of dynamic range, qPCR has a wider dynamic range, typically 6 to 8 logs, while the digital PCR has a narrower dynamic range. And this is because the digital PCR, it does not rely on the standard curve, but also it counts the true copies of the DNA or the oligo in your samples. For the reproducibility, the qPCR is good, but again, can be affected by the efficiency drifts and curve quality. While the digital PCR is excellent, has an excellent inter-run and inter-lab reproducibility. For the data analysis, again, the qPCR, right, it relies on external standards. So those standards needs to be prepared like correctly and accurate in order to obtain good data. While the digital PCR, it measures the true copies. So in here on the right side, I have 2 pictures of the digital PCR of some samples that we have run on Amplato. The top one is the negative control, and the bottom one is our positive control. So this slide here shows how the workflow compared to ELISA. So on the ELISA, we start with plate coding and then blocking, and then comes with sample incubation. Then after sample incubation, we have some washes, and then we add our detection antibody, which is biotherapeutics. So in the normal ELISA, we would come with the conjugate and then add the substrate and read the plate. But in here, we stop right here, and instead of adding the conjugate, we add the nanoparticles, incubated for a certain amount of time, and then we release the copies of DNA from the particles, and then used that samples to analyze in a qPCR or digital PCR. In terms of applications, Amplato support a wide range of applications, including HCP monitoring, biomarker quantification, PK assessment, and also immunogenicity. The technology enables accurate measurement of low abundant analytes that traditional ELISA and ECL methods often fail to detect. And we're going to demonstrate that in a few cases, few examples that we're going to talk today. In particular, like in PKA, it provides high sensitivity and larger dynamic range. In biomarker assays, when the biologic being tested. It downregulates cytokinase. So ultrasensitive technology is really important for that, because then you have a decrease in concentration in there. And also in microsampling. So those analyses, they don't require large volume for the sample. So it's really important there in when you're doing microsampling studies. With that, I'll pass the stage to Jesse, which is now going to talk about the PKA and HCP analysis.
So, Jesse, before you jump in, I just wanted to remind everybody that if you have any question, I do already see 1, 2 questions. So anybody else have questions, feel free to submit it. You can submit it throughout the session. All right, go ahead, Jesse.
Sure. Can you hear me? Here we go. Apologies, we are in the same room, so just making sure the sound is coming through properly. All right. So Amplato to support pharmacokinetic quantification. So at this stage, it's important to step back and consider why ultrasensitivity is so critical in bioanalysis. Ultrasensitivity detection isn't just a technical consideration. It is becoming more of an increasingly regulatory expectation. The current FDA and EMA guidance, including the ICH-M10, stresses that the PK data must be generated using validated assay systems with sufficient sensitivity across the entire concentration time profile. So many biologics, biomarkers, and process-related impurities exist at extremely low concentrations, often below the detection limits of the traditional ELISA or ECL platforms. For low dose or high potent biologics, concentrations at later time points may approach or fall below these limits. When this happens, the data can be masked in the sensitivity, making the PK parameters like half-life and clearance rely more on extrapolation. And regulators expect such limitations to be minimized wherever scientifically possible. So this particularly critical is when you're analyzing low-dose biologics, downregulated biomarkers, or late PK time points. In these cases, sensitivity is not optional. It directly affects the data quality, interpretation, and the regulatory confidence. So in the case of Amplato, it can extend the PK visibility into the terminal phase, enabling quantifiable data at late time points. And this reduces the reliance on that extrapolation and strengthens the scientific justification of the PK parameters, which is particularly important during regulatory review. So romiplastim as a case study. So romiplastim is a great example where the sensitivity really matters. It's a low abundance fusion protein, and traditional ELISA methods struggle to reliably quantify concentrations below 10 picogram per mil. However, those low concentrations are exactly where we need the data to be able to fully characterize that PK behavior, especially at the late time points in clinical studies. So for the romiplastim assay designs, so we took the romiplastim and we evaluated it across multiple platforms. So here you can see the differences in these assay designs. At a high level, the key distinction across these platforms is the detection system. In all cases, the romiplastim is captured using the TPO receptor and bound by a specific anti-romiplastim antibody, followed by an anti-species detection antibody. The point where the assays really kind of diverge is at the detection conjugate. And you can see that here on these three. Sorry, Will, you have my mouse. And so what ultimately drives the sensitivity are these different signal amplification strategies. ELISA is using an HRP-based colorimetric readout, where the MSD relies on the electrochemiluminescence from the sulfo tag, and Amplato converts the detection event into DNA signal that is then amplified separately in a PCR. So for ELISA and MSD, signal generation is limited by how much conjugate can physically be attached to the detection antibody. And in contrast, Amplato uses the nanoparticle-linked DNA that can be further amplified outside the plate, enabling substantially higher sensitivity. So in these next slides, we have some data just regarding these three platforms. With ELISA, we achieve sensitivities around 10 to 20 picogram per mil. Here you can see the sensitivity was limited to approximately 10 picogram per mil, which is restricting the detection at later time points if we were to go into a clinical study with this as our analytical method. For the MSD, which is on the next slide. Oh, we're on there. Okay. So you can see the MSD has improved sensitivity around 2.5 to 5 picogram per mil. And so what you can see here is while the QCs have been recovering at 2.5 picogram per mil, we were unable to get the sensitivity passing at this level. So that was able to be achieved at around 5 picogram per mil. So truly the sensitivity is more likely to be the 5 picogram per mil, whereas the QCs prepared in the same matrix as the calibration curve does look like it can achieve the 2.5. We are more calling this sensitivity at around 5 picogram. So for the Amplato, we were able to achieve an analytical sensitivity of approximately 1 picogram per mil. This represents a clear step change in performance and demonstrates how ultrasensitive platforms enable more complete PK characterization. And on the next slide here, you'll be able to see that unlike the MSD, the selectivity was able to meet acceptance criteria in 10 individual serums, with the 11th serum listed here, a pool of all of the serums, which clearly indicates the sensitivity in multiple individuals without matrix interference. And from a regulatory standpoint, this improvement translates to fewer below limit of quantitation samples and improves the confidence in the PK parameters and stronger support for the dose justification assessment. And so finally, on the last slide regarding romiplastum, this comparison highlights the ultrasensitivity. Amplato extends the PK visibility, improves the confidence near the LLOQ, and strengthens the conclusions around the half-life, clearance, and biosimilarity. And you can see the tenfold increase in the sensitivity between the traditional ELISA and the Amplato here. Moving on to our next case study. So for Amplato, to support the HCP detection and some regulatory expectations surrounding that. So HCPs are considered critical process-related impurities, and regulatory agencies increasingly expect sensitive and specific methods capable of detecting individual HCPs at very low levels. Individual HCPs can remain in the final drug substance at tens to hundreds of picogram per mil levels, where traditional ELISA and ECL assays often struggle to kind of achieve those levels due to background and limited sensitivity. Guidance from the FDA and the EMA emphasize that the residual HCPs, even at these low picogram per mil levels, can pose a risk to the product stability and the patient safety. And so in this context, Amplato demonstrated significantly improved sensitivity for the capsaicin D compared to the colorimetric and ECL methods, aligning with the growing regulatory focus on sensitive product-specific HCP monitoring. And so before we get into that data, just wanted to recognize where some of this has been generated from. So Somro has been awarded an UK Canada Biomanufacturing Award for for a project focused on real-time analytics to support biomanufacturing. So this work that I'm about to share is part of an international consortium developing near real-time process analytical technology platforms integrated with AI-driven data analytics with the goal of modernizing biologics, manufacturing, and strengthening pandemic preparedness. As part of this initiative, we're exploring ultrasensitive HCP detection strategies to better the biomanufacturing process. And the next few slides will just discuss the ultrasensitive analysis. So for the cathepsin assay design, similar to romiplastum example, we evaluated this across multiple detection platforms, including ELISA, MSD, and Emplato. And then for Emplato, we actually assessed with the two readouts that Will had previously discussed with the qPCR and the digital PCR readouts. And that's fine. So what this data shows is pretty clear, similar to the romiplastum, in a stepwise improvement of the detection capability as we move across the platforms. So here, using the traditional ELISA, the LOD was approximately 1,000 picogram per mil. And then on the next slide, we were able to see in the MSD a twofold increase to achieve around 50 picogram per mil. And then when we applied the Emplato with the qPCR readout, we were able to drop the LOD to around 125 picogram per mil. And then for the digital PCR, we were able to achieve a final sensitivity of 31.25 picogram per mil. And here you'll see that the concentration is listed here twice, once with the picogram per mil, and then once is actually the DNA copies per microliter. And then in this instance, this is actually 1 microliter, so this is number of copies per well. You would be able to increase the sensitivity potentially by increasing the volume. So we may be able to get into those tens levels of the HCP detection. And so then qPCR overall represents a 8 to 4-fold improvement in sensitivity, whereas the digital PCR, we were able to get about a 32-fold improvement compared to the traditional ELISA. And just as a note, importantly, this level of sensitivity isn't just really about advancing the technology for its own sake. It's more that the regulatory expectations are increasingly being required to the detection of specific low abundance HCPs in these complex matrices. And as purification processes become more efficient, only ultrasensitive technologies will be able to reliably meet these requirements. And back to you, Will.
All right, thank you, Jesse. Now I'm going to talk about the Emplato for biomarkers, especially talking about multiplexing. So multiplexing is a topic that has come up many times in our previous presentations, and we've decided and we've received a lot of interest and questions around it. So in response to that, we wanted to share some background on the multiplexing and also highlight that what we have accomplished so far using Emplato. So for biomarkers, as I was mentioning earlier, in preclinical and clinical studies, therapeutical interventions often don't regulate inflammatory cytokines such as TNF-alpha and IL-6 to very low to near baseline levels. So ultrasensitive detection technologies are therefore critical to accurately quantify residual cytokinase levels and distinguish true pharmacodynamic effects from assay noise. Also, multiplexing in here enables simultaneous measurements of multiple biomarkers from minimal sample volume. So in this case, when you're doing preclinical clinical studies, you don't need to reserve two aliquots to analyze those two biomarkers. So in this case, you just need one, providing deeper biological insight and greater efficiency and consistency. So to achieve this multiplexing with Amplato, we have designed two distinct nanoparticles, each carrying a unique oligonucleotide sequence. These distinct oligonucleotide allow each analyte to be independently identified and quantified within the same assay. So in here, I'm calling them marker A and marker B. The amplification for those oligonucleotides now, they are performed using a probe-based approach, enabling a clear signal discrimination between the targets and minimizing cross-reactivity. This design is optimized. We optimized them for sequence homology, so the primers, they do not crosstalk with the sequences. And also we have optimized all the reaction conditions, in this case, the primers and probes. This design is optimized, like I said, formally and support robust, reliable multiplex detection. And in here, I have the amplification curves for these two biomarkers. So on the left, I have the amplification, and on the right side, we have the kind of the calibration curve. And you can see the efficiency in there is pretty good. And we are able to detect both at the same time. So in terms of workflow, here for the multiplexing, we start with the plate coding. In this case, we have a cocktail of coding antibodies that are targeting both IL-6 and TNF-alpha. And then after blocking, we come in with the samples. While the sample is incubating, so we incubate the detection antibody, which is biotherapeutics. So the top one here, let's say that is towards the IL-6, and the bottom one is towards the TNF-alpha. So we incubate them with the specific nanoparticles for a certain amount of time. And then after that, we incubated on the plate after the sample incubation. We leave that for a specific amount of time, and then after some washes, we release the DNA from those particles, and then we analyze with qPCR or digital PCR. And in that way, we achieve that multiplexing. So for the assay design for the IL-6 and TNF-alpha, so they are demonstrated here. You guys are already familiar with these formats. Jesse has demonstrated a couple of them. But so in here, we have the plate coded with anti-IL-6 and anti-TNF-alpha, where these target bind to specific coding antibodies, and then we come with the complex that is formed in the solution phase, right? We incubated previously the marker A with the IL-6 and marker B with the TNF-alpha. So they form this complex, and then we release the DNA and quantify using qPCR or digital PCR. So our initial approach was to develop each individually, like the TNF-alpha and the IL-6. I'm not going to go into much details on how we achieved the sensitivity. Jesse explained a little bit on the pKa and on the cathepsin D that on Plato you can achieve higher sensitivities compared to ELISA. And here I have just a summary. So for the IL-6, the ELISA, the LOD was about 37.5 picogram per mil. With the Amplato, we achieved around 2.3 picogram per mil, which is about 16 times higher. And for the TNF-alpha, the ELISA had an LOD of around 150. For Amplato, we got it down to 9.4, which is about 16 times higher as well. So our next steps will be combining these two assays into that multiplexing system. and also expanding to more molecules, more cytokinase. There we are kind of limited based on the amount of fluorophore that we have, the panels that we have for the PCR and the qPCR and digital PCR. So the idea is to initially develop in the qPCR, and then we translate that in the digital PCR. So in conclusion, the platform supports a broader range of applications, like we have been talking, from biomanufacturing and CMC to routine analysis, including PK, biomarkers, and HCPs. Immuno-PCR enabled by Amplato technology provides ultrasensitive detection from picograms to sub-picogram levels, expanding bioanalytical capabilities beyond traditional ELISA and ECL methods. Despite its advantages, the immuno-PCR remains underutilized across bioanalytical and CMC applications due to the challenges that I mentioned at the beginning of my presentation, such as instrument compatibility, nanoparticle reproducibility, trained personnel, and the lab itself, right, which are not designed to run immuno-PCR, but rather just ELISAs. So we were able to overcome these historical limitations by using the nanoparticles combining with optimized method execution. So we have segregated some areas of our lab, including we have opened another lab where we're doing our analysis in there. And also looking at the reproducibility of the nanoparticles and the quality of them. So for the HCP, Amplato address critical gaps as well in HCP detection, like Jesse was mentioning, where up to 30% of the HCPs can be undetectable by conventional assays. And finally here, this technology is well suited for emerging modalities, such as cell and gene therapies, viral vectors, ultra-low dose biologics, and highly purified monoclonal antibodies. With that, I would like to thank all Samru team for all the support and also great work, and all of you as well for being here, listening to our talk. And we'll be happy now to answer any questions.
So, and you have our email addresses here. Also, feel free to send us the questions if we cannot get through all of them, but we do have some time here. Maybe I'll kick it off with the first one. I think this person did get the response, but I'll ask it anyway. Is the whole immunocomplex enter digital PCR partition or only the DNA portion?
Yeah, so it's only the DNA portion. Let me go back in here, like I was mentioning at the beginning, on the workflow. Just a minute. Yeah, so after we incubate the particles on the plate, which, like I was mentioning, can be either an ELISA strip or a PCR plate. So we wash any unbound particles, any complex that's not formed. Then we release using heat. So heat release those particles, and then we insert the samples into the qPCR, digital PCR. The volume that we usually use is 5 microliters. For the digital PCR as well is 5 microliters.
Yeah. Thank you, Will. I see a couple of people maybe raising hand. I would encourage you, please go ahead and submit the questions on the question Q&A panel of this of the seminar. The next one Well, I'll let you guys choose whoever wants to take that question. How does the dynamic range compares with MSD and Simoa?
Okay. I can take that. Can you hear me through Will's mic?
Yep, we can hear. We can
Okay.
hear.
Perfect.
Yeah.
Just in general, Amplato is going to have a wider dynamic range than the traditional ELISA or MSD. This dynamic range does depend primarily on the number of DNA copies on the nanoparticle itself. More copies would lead to a higher significant range. And then also back to what Will was kind of saying at the beginning, where which type of readout system that you wanted to use. qPCR is going to have a wider range, whereas digital is going to have ultra sensitivity and more sensitivity, but it will have a lower range. So you're going to want to use that when you are really just targeting kind of a positive negative on a really low abundant protein.
Yeah, and just to point out that in Amplato here, every single binding event is associated with 200, 300 copies.
Mm-hmm.
So that's how we achieve like really high sensitivity. So for the dynamic range, again, it is wider than the Simoa, but also it's limited based on the qPCR, the instrument, the qPCR or the digital PCR. So the digital PCR is really sensitive, so you're going to have a shorter range in there, while the qPCR provides a broader range.
Thank you both.
I see another question in here, Vic. Sorry, it's do you have nanoparticles functionalized with other molecules besides strep? And could you quantify two proteins at the same time? So like I mentioned, yes, we can do multiplexing, but we like to keep a streptavidin because of the versatility of that. We can conjugate directly into the particles, but that involves more work, because then if you make one particle conjugated with a specific detection antibody, so that's it, right? You cannot, it's not versatile. You're not going to use it to detect other types of proteins or biologics, but you can conjugate directly.
Right. Thanks, Will. I think there are a couple of questions on the Simoa, and maybe I can take on that one. So we actually never did an head-to-head comparison with Simoa, as we do not have the Simoa platform in-house. But I know that we have clients who are using Amplato platform, and they have access to Simoa. Some of the biomarker assays that we have performed, and this is more of an indirect comparison, I think the sensitivity, depending on assay, it will be very comparable. It's not a little better, but also in terms of dynamic range. And again, it varies. I think Simoa can do 3 to 4 logs and sometimes maybe 5. I think with immuno-PCR, if you have the right design, you can go up to 4 to 6 log. And as Jesse and Will both mentioning, it's dependent on the number of copies and the endpoint qPCR versus digital PCR. qPCR, maybe you can do a little more broader dynamic range. But we have actually never compared directly on the same assay head-to-head with Simoa. I think, Will, you want to take on this one with are the biomarker testing conducted in the same well? I think the person asking question, you would have strep NP DNA target 1 competing with NP strep DNA target 2.
Yes. So they are both conducted in the same well, but they will not crosstalk because we have previously incubated the particles. Let me bring here the workflow here. So we have previously incubated the detection antibody with a specific particle. So in this case in here, for us to avoid this crosstalk, the concentration for the detection antibody needs to be limiting. So what means that all the detection antibody needs to be captured by the particles. So in this case, even though if you have an excess of particles in there and you add it into your sample, so there is no more detection antibody to bind it to it, so you're not going to have that cross-talking. So the signal that you're going to get is specifically from the IL-6 or the TNF alpha.
And I mean, with this one, we have, if I may add, multiple options. We could put other tags. So other than biotin strep, we do use DIG, anti-DIG quite a bit. So there are different ways you can execute that. I see a few more questions. Keep them coming. We do have some time, so we'd be happy to answer those. And I think, frankly, that's the exciting part of for me is that be able to interact and answer those questions. I'll just go in order, so it may sound a little random here. I'll let you, Will, Jesse, you guys decide and take on that. Next one, typical time in M-Plateau, qPCR, dPCR versus ELISA, MSD from sample prep to getting the final data. So I think this is a very relevant slide you have in front of you. Okay, go ahead.
Yeah, so it all depends on how you have your assay automated. So you can have Plato on the assay execution being the same time as the ELISA. Full sample, but yeah.
So like to add on to that, really the only additive timing is like really just the running of the PCR.
Yeah, because the data analysis, you also do the same thing as you would do with the ELISA. So do a calibration curve regression, checking QCs and so on, back calculated.
All right, thank you. So the next one, can the immuno-PCR improve overall specificity of the method? Or is it still driven by the selection of antibodies used in the primary assay? Very good question.
Yeah, very good question.
We talk about it quite a bit. Go ahead.
Yeah. And so because Plato, right, is a ultra-sensitive platform, so the quality of the detection antibody is really important. So the specificity, it will not improve the specificity. It will come from the pair of antibodies that you're using. But the detection antibody quality, it needs to be good because if you have cross-talking with, if you're coding or if you're blocking, then those particles will be immobilized by the detection antibody, and then you have background issues. And you have seen this quite a lot with the traditional ELISAs when you are adapting, because traditional ELISAs, they use high concentration of the reagents, and in Plato, you don't need to use that high concentrations. And so when you're using high concentration for reagents, the specificity is important, but is not so pronounced on your background as it is in the Plato.
One thing I would add, Will, is that if you have the M-Plato in this platform, we use probably half, if not 25% of the antibody that you would use usually in an ELISA platform. So usually much lower. So if you have non-specific, that's one way it may help. I don't think we have a direct data on it, but I think one way it may help with specificity is that if you have a polyclonal detection antibody and it's not very specific, you are using so little that you might be getting less non-specific binding. But there is no bottom line is that you do need quality antibody. But you can get away with, maybe even some low affinity antibody just because you don't need a strong binding. You just need enough binding so that, you can amplify And if I recall your first slide, Will, you have somewhere between 200 reporter DNA
283. Yeah.
per particle. So if you are putting this many reporter, you don't really need strong binding. A lot of times we run into specificity issue in ELISA, MSD, or any immunoassays that we need to put more antibody to increase the signal. And the with more antibody, you get more non-specific binding too. So the amplification that we are doing here, compared to traditional immuno-PCR, in traditional immuno-PCR, you might be putting 1, 2, 3, maybe 4 reporter per DNA. In this case, through the use of nanoparticle, we can actually load those particles with 200 to 500 oligos. So that's a significant, amplification. Okay. Next one, validation of M-Plateau assay used for regulated bioanalysis for PK or biomarker. If biomarker, what panel? If PK, what drug modalities? So I know that we have worked with multiple clients to submit. A lot of the work has been exploratory, but we have done, at least I can think of one fusion protein, one monoclonal antibody, and the other one is a peptide therapeutics, really. For the peptide therapeutics, I actually presented that. I know we've been working on this quite a bit. I feel like it's maybe 2017 or 16 Land O'Lakes conference. We presented some data that was submitted for the regulatory decision making. For biomarker panel, not probably the panel. Will you have done the individual biomarker like C5?
Yeah. C5a,
Mhm.
TNF alpha, IL-6.
But we haven't submitted any multiplex work for any regulatory decision making, just to make the answer complete here.
And just to be clear, like those IL-6 and TNF alpha data that I demonstrated in here, they are with the new particles, the marker A, so the probe-based approach.
Yeah. Maybe the last questions, is that maybe I know, Will, you mentioned it on the slide, but can you elaborate on some of the operational challenges? I think you were saying about the lab compatibility or reagent. Can you elaborate on some of the challenges from the operational perspective?
Yes, so like I was mentioning, there are some equipments that they are not really designed for immuno-PCR. So even on the PCR itself, right? Sometimes like the pipettes that we have multi-channel is not usually with the volumes that are required to run those samples. So we have challenges in there, and also in the lab space itself. So it's really easy because those technologies, right? They are sensitive. So qPCR, digital, they are really sensitive. So because of that, they're also really easy prompt for contamination. So if you doing your assay and also doing your PCR mix preparation on kind of same area, you're gonna have contamination, and then that contamination is not really gonna come from your it's gonna affect your standard curve, right? So the background, you're not gonna get the true background that is coming from the assay, but rather from just contamination with the pipettes, and the environment itself. So you need really some laboratory personnel that are trained with cleaning and running PCR and also running the ELISA.
Okay. Thank you, Will. I'm just looking through all my notes. I think, Will, if you can go to the last slide. Just wanted to give people a quick view of our contact information. If you can think of any follow-up questions, it may come in. It may come up. If you just want to share your experience, I think going back to the last question, when we try to provide reagents to some of our clients to execute this assay in their lab, we actually have a pretty mixed result. And some of the challenges are that a lot of those labs are designed for ELISA type of assays, not quite immuno-PCR. So if we actually offering this more as a service because of those challenges, and some of our clients, they would prefer instead of making all these changes in their lab, to have us do it. But if you are also looking into more of a long-term implementation in your lab, as opposed to doing one assay, it may not make sense. But if you're looking long-term implementation, this team will be available for any sort of technical discussion, share some of our pain points, and hopefully we all learn from it. So with that, I would like to thank our speakers. I would like to thank all the attendees for attending this exciting seminar. And I would like to thank AAPS for providing this platform to share our work. Thank you, everyone, and you guys have a good day.
Thank you so
Thank
much.
you.